Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Hearty welcome to JnanaMarga URA classes. I am G.K Bhat taking a biology class.
Good evening, sir.
Very good evening. Yeah. I hope the board is visible to you. I am sure you are aware the corona pandemic outburst again, what is called a third wave. I request you all to take extreme care and see that we are safe. Please you wear the mask whenever you are coming in contact with the people, and also keep at a safe distance. These are the two things. Mask is just to prevent any droplet entering into your nose, and the same thing is true with regard to maintenance of the distance. These are the two important things to keep away from the viruses. Viruses will not fly in the air and come to you, especially this COVID-19 virus. So let us be always on the safer side. Coming to this particular chapter, you know that the biotechnology is a known branch of science to each and everyone nowadays, because we are making use of the product of biotechnology in our day-to-day life. In these 2-3 classes, we shall look into what are the principles and processes involved in biotechnology. Biotechnology deals with the techniques of using living organisms or enzymes from organisms to produce products and processes that are useful to humans. Always ask a question in the board examination also. What is biotechnology? Biotechnology, where living organisms are used or enzymes from organisms are used. Why for? To get the products which we want, in which we are interested. In this sense, making a curd, you already know, lactobacillus bacteria is involved in it. Making bread or wine, we have already learned, yeast converts the sugar to alcohol and carbon dioxide, which are actually just byproducts of the biochemical process. Yeast uses to extract energy from sugar, which are all microbe-mediated processes. Milk becoming curd or production of the alcohol, getting bread, all these are the products of biotechnology, or they are all microbe-mediated processes. And these are also considered as part of old biotechnology. Now we may not give the name product of biotechnology to these reactions which we are seeing in our day-to-day life. However, when biotechnology is used in a restricted sense today, that is, to refer to such of these those processes which use a genetically modified organism. Always ask a question, what is a GMO? Genetically modified organisms, to achieve the same on a larger scale. This is referred as a modern biotechnology. The same one or the other product in which we are interested, we are getting this product in a much more big or large scale. And that is what generally referred to as modern biotechnology nowadays. Further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube, which you have already studied under human reproduction chapter, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. So, let us look into more details about this. The European Federation of Biotechnology, again important in this abbreviation EFB, European Federation of Biotechnology, has given a beautiful definition of biotechnology that encompasses both the traditional view and modern molecular biotechnology. It includes both old biotechnology and the modern biotechnology. The definition given by European Federation of Biotechnology is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. This is what the definition given by EFB. The integration, we are combining all this, the integration, natural science and organisms. Which are the natural sciences? Kanisa? Kanisa?
Yes, sir.
What do you mean by natural science? Or anyone else? Who will tell me what is the natural science? Hello? Namrata? Navya? Prithvi? No idea? Hello? Blessy?
Sir,
Who is telling? Yes, come on.
sir, a branch of a science which deals with the physical world.
That means which science comes in this? The integration of natural science and organisms. Is it that you saw that in internet?
Or is it environmental?
Natural world, sir.
I want you to tell me. I shall give you the time. Please think about for some time. You know, there are different sciences. You must know what is natural science, what is physical science, what is social science. Okay. The integration of natural science and organisms, cells, parts thereof, and molecular analogs. What is that molecular analogs? Compound having a structure similar to that of another compound. For the products and services is what definition that is given by EFB, European Federation of Biotechnology. Therefore, if at all you are asked to define biotechnology, better give this definition in the examination, board examination. Now, let us look into principles of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology are: one, genetic engineering; two, chemical engineering. As you can deduce, genetic engineering involves techniques to alter the chemistry of genetic material. You know, genetic material is the DNA and RNA. So, genetic engineering involves the technique which alters the chemistry of genetic material to introduce these into host organism and thus change the phenotype of the host organism. What is the principle here? Genetic engineering involves technique to alter the chemistry of genetic material. So what happens? If the chemistry of DNA and RNA, what do you mean by? What do you mean by altering chemistry of genetic material, chemistry of DNA and RNA? What do you mean by that? What do you mean by that? genetic engineering technique, it involves a technique where chemistry of genetic material, DNA and RNA is altered. Siddesh, what do you say? Vishwas, Rakesh, come on. Rafa, Aisha,
Sir, is it altering their structure?
how? By what? You are right. You are right. Altering what structure?
Altering DNA sequences.
That means, what is the DNA sequence? Sequence of bases. So, addition, deletion, substitution. Yes, you are right. It is by altering the chemistry of genetic material means we are going to alter the basis sequence of DNA and RNA. And after this, introducing this altered DNA or RNA, so modified DNA or RNA, introducing it to the host organism. And always the phenotype of an organism is decided by genotype of the organism. Am I right? Am I right? You know about that. So in this way, we are going to change the chemistry of genetic material, chemistry of DNA and RNA, and introduce this into the host organism, and thereby we modify the phenotype of the host organism. This is what is called genetic engineering. I hope it is clear. Is it clear? Harshavardhan, Viswita, Nadira.
Yes, sir.
Okay. Now let us look into chemical engineering. I don't know why that Harshavardhan, Siddesh, you people are not talking, not replying. Now let us look into
Yes, sir. Understood, sir.
Okay. Now let us look into chemical engineering. Chemical engineering, this deals with maintenance of sterile ambience. You must know here, sterile means microbial contamination free. They are free from any contamination by any microbe. Maintenance of a sterile ambience. Ambience means atmosphere. Ambient temperature means temperature of the atmosphere, temperature of the environment. Maintenance of a sterile ambience in the chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in the large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. This is what you say chemical engineering. So, please know, chemical engineering deals with the production of antibiotics, vaccines, enzymes, etc. in a microbe-free condition. Whatever we desire. So in this way, you have genetic engineering and chemical engineering. I hope it is understood and we can go ahead. Right. Let us now understand the conceptual development of the principles of genetic engineering. What are they? You probably appreciated the advantage of sexual reproduction over asexual reproduction. Am I right? You have studied about the sexual and asexual reproduction. Who will tell me why we appreciate that sexual reproduction is more advantageous than asexual reproduction?
Because recombinants are formed.
Very good. Because of the recombination of the genes. So the former provides opportunities for variation. Sexual reproduction gives the opportunity for variations and formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. So asexual reproduction preserves the genetic information. What do you mean by that? What do you mean by that? Asexual reproduction preserves the genetic information while sexual reproduction permits variation. You have already heard variation is due to because of the crossing over taking place during the gametogenesis. So you do come across variation. In sexual reproduction, we come across variation because sexual reproduction invariably involves the meiosis, wherein we come across crossing over recombination of the genes. So in what way? Asexual reproduction preserves the genetic information. What do you mean by that? What do you mean by that? Asexual reproduction preserves the genetic information. Nadira?
Offspring are clones.
Come on. Yeah. Who said it?
Kenisa.
Kenisa, you are right. In asexual reproduction, it is always uniparental. When asexual reproduction is uniparental, what do you see? Whatever the genetic information is there in the parent, systematically that comes to the progeny unaltered. Okay, so that is what asexual reproduction preserves the genetic information, right? Traditional hybridization procedures used in the plant and animal breeding. Traditional hybridization procedure is what? You are just crossing two of the organisms and getting a hybrid. Whatever the two varieties you want, the so-called best varieties for one or the other particular character, you are crossing the two and you are getting a hybrid. Traditional hybridization procedures used in the plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. You understand this? Have you understood the meaning of this sentence? Whenever we just cross-pollinate two of the plants with the desirable character, don't you think that along with the desired genes, undesired genes also come? There is no way to prevent it. Blessy, have you understood this? Aisha, Rafa,
Yes, sir.
Tanusri, Jayasri, have you followed this? Whenever we cross two organisms, to bring the desired genes together, along with the desired genes, undesirable genes also come, which we cannot prevent. I hope it is understood, Tanusri.
Yes, sir.
Right? So the techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning, and gene transfer, overcome this limitation. In this way, genetic engineering is more important. What happens here? No doubt, the technique of genetic engineering involves creation of recombinant DNA, use of gene cloning, and gene transfer. This overcomes this limitation. What this limitation? Siddesh, it overcomes what? Siddesh,
Sir,
it overcomes what?
variation.
Pardon? I am asking you, by genetic engineering, which includes recombinant DNA, use of gene cloning, and gene transfer, I said overcomes this limitation. What limitation? Who will tell me? It overcomes what?
Multiplication of undesirable genes.
Yeah, multiplication of undesirable genes. What I said, I am just now showing you in what way genetic engineering is different from that of typical hybridization. In a typical traditional hybridization, Siddesh, please listen to the lecture carefully. In a
Yes,
typical
sir.
in a typical traditional hybridization, whenever we cross two organisms, whenever we are going to get hybrid, the hybrid definitely it may show desirable genes, but at the same time, it also shows undesirable genes. It also brings about undesirable genes. Now let us think that one particular, just a tentative example. I am just giving one example. Hypothetical. And it need not be actually true. Now you are crossing one very good variety of your cow, local cow, with a very good variety of high yielding breed. You are crossing two cow. a local breed of cow is crossed with a breed which is known to give very good milk yield. Now what happens? In this hybrid, you may get the hybrid to develop that very good amount of milk yield, but at the same time, it may also get another particular habit. That is, whenever one goes near to that to udder the milk, to get the milk from the udder, it is a very bad habit of just kicking. It starts kicking. Tanusri, Jayasri, Savitri, have you not
Yes,
seen,
sir.
have you not seen some cow when you go to udder the milk, when you go to take the milk, it starts kicking?
Yes, sir.
If at all, is it that we are going to have this hybrid to get the kicking cow? No. Simply I said, but the genetic engineering, what it does, it overcomes that coming of undesired genes with the desired genes. So it overcomes this limitation and allows us to isolate and introduce only one or a set of desirable genes. In the genetic engineering, therefore you must remember, in the genetic engineering, it enables us to isolate and introduce only one or a set of desirable genes without introducing undesirable gene into the target organism. This is what introducing desirable gene is known as recombinant DNA technology. In recombinant DNA technology, the desirable gene or genes are isolated, cloned, and transferred to the target organism. So if at all you are asked, what is recombinant DNA technology? Answer is, the recombinant DNA technology is the process in which the desirable gene or genes are isolated, cloned, and transferred to the target organism. Now let us try to understand this particular, definition. What do you mean by this? Okay, please read this. Whatever I have given in the red color, reading, you please read this and one of you come forward to explain me this each and every word in this sentence. Who will do that? Who will explain? Is there anyone who can come forward to explain this sentence to me? Word by word. Hello, who will tell me? Janani, are you there?
Yes, sir.
Can you? Jyoti, Namrata, Prithvi, Lohit. Lohit, are you there? Lohit,
Yes, sir.
who will tell me? Who will explain this particular sentence? This is very meaningful sentence. Come on, quick, I will help you. Hello? I shall help you. In the recombinant DNA technology, the desirable gene or genes are isolated. I hope you know. What do you mean by cloned? Anyone knows about this?
Copied, sir.
Yes. Yeah, who is this?
Rafa.
Yeah, Rafa, tell me.
Made similar copies
Yeah, make similar copies. Why you should make similar copies?
to transfer it.
To transfer more number of cores. So it is cloned and transferred to the target organism. Who is the target organism? Who is the target organism? Rafa,
We are supposed to select that.
we?
We will select that to the one we want to change. That is the target organ.
Yeah. The target organ is the one in which we are interested to transfer the desirable gene.
Change.
Yeah, that is the target organ. We wanted to take the desirable gene from, now let us see that X is one organism, Y is another organism. We wanted to transfer gene from X and we wanted to give the gene to, put the gene inside the Y. So target organism here is Y. And we isolated the genes from X. After isolating genes, we are going to multiply the copies of the genes by the process of cloning. Now let us study this. Do you know the likely fate of a piece of DNA which is somehow transferred into alien organism? What happens? If you just take a piece of DNA, there are techniques, and you are introducing this DNA into one organism, somehow you just to do somehow you are transferring this DNA into another organism, alien organism. What will happen to this DNA? What will happen? This DNA, whatever you are putting a small piece of DNA from say X to Y, it may show some impact in Y, but it will not pass to the offspring of Y. I hope you understand this. Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organism. You may see this DNA in that particular organism. It may show to certain extent some impact of this addition, but it will not pass to the progeny of the organism. But when it gets integrated into the genome of the recipient, what do you mean by genome? But when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. What is the genome? What is genome here? What is genome? Genome is a
DNA.
nothing but a DNA. It is nothing but DNA. But when it gets, you are taking a small piece of DNA, you wanted to transfer it to another organism, you are not directly transferring it into another organism. You are ligating it or tying it or putting it in the DNA of the recipient. When you just put inside the DNA of the recipient, it may multiply and be inherited along with the host DNA. Along with the host DNA. Host DNA is the here target DNA. This is because the alien piece of DNA has become part of a chromosome which has the ability to replicate. Understood? Is it clear? In a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating replication. I hope I request you to recall the replication. Okay? Semi-conserved method replication. And you know what is happening during replication? Replication of the DNA results in a duplication of the chromosome. So, in a chromosome, there is a specific DNA sequence. In a chromosome, there is a specific DNA sequence. That means in the DNA of the chromosome, in the DNA of a chromosome, that is a part of DNA. In a chromosome, there is a specific DNA sequence called ORI, origin of replication, which is responsible for initiating replication. Right? I request you, if you don't follow any of these, you please stop me and interrupt me and ask me if it is a doubt, and let us go to the next one. Now, therefore, for the multiplication of any alien piece of DNA, any alien piece of DNA means any DNA which you have chosen to introduce into the host, Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be a part of a chromosome, or it needs to be a part of the DNA, which has a specific sequence known as origin of replication. So, we are adding in that region where you would come across specific sequence of gene, and this particular sequence is known as the origin of replication. Thus, an alien DNA is linked with the origin of replication. Origin of replication is a part of DNA. An alien DNA or a foreign DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called cloning. What is that? Multiplication. DNA multiplying in a large number. This is called cloning, or making multiple identical copies of any template DNA. Template DNA is a DNA which is used for replication. Is it clear? Savitri,
Yes, sir.
Tanusri,
Yes, sir.
Jayasri.
Sir, cloning you explain once again.
Which one?
Cloning, sir.
I did not hear you.
Cloning, sir.
Yeah, cloning is nothing but multiplying. Instead of having only one piece, you are going to have 100 pieces. We are going to multiply it. So, if at all, now you just listen to me. You have one DNA. This is the DNA. Okay, into this DNA, I want to add this DNA. Okay, you are seeing this DNA. What do you call this DNA? What do you call this DNA now?
X.
Alien piece. Foreign
Alien
piece. Foreign piece, alien piece. In this DNA, you are interested. You wanted this particular segment. I am showing this particular segment of DNA you want. You want to put where? You want to add this particular piece of DNA into this, my another hand you are seeing here. And
Yes,
to this DNA, you wanted to add, this is a target organism. This is the target one. You want to add this DNA into this DNA, into the another DNA. But this DNA, you want to add it to the another DNA. So this DNA is multiplied. Yeah, this is the, I'll come to that point again, and there are different terminologies and all. I'll come to that again. You are not going to just put one particular piece and add it to this and keep quiet. No, you are not going to keep quiet with only one piece of DNA added to the host organism. You want to add a number of pieces of this, whatever the small pieces, etc. You are not going to add only one piece. You are going to add hundreds of pieces into the host organism. Can anyone tell me why? Why we are going for 100 of pieces, not only one piece? Hello?
The desired gene will be available.
So here, okay, that's not the answer. I'll come to that. An alien DNA is linked to the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. When you add to this piece of DNA, when you add to this host organism, you are not going to keep quiet with that. You want to see that this host DNA and this alien DNA, both these, they are going to multiply in a large number before you process further. Okay, let us see that. This can also be called cloning. But remember, Jayasri, cloning means making a number of copies of the same. You just remember now, I will come to that point again. I will explain better in another step. This can also be called cloning or making multiple identical copies of any template DNA. Making multiple identical copies of any template DNA. What is that template DNA? Why that we are going to have multiple identical copies of a template DNA? What you call this template DNA otherwise?
Nonsense DNA.
No, don't go to that extent, okay? Otherwise, here, what you call template DNA otherwise? Here only, whatever the example I have given. Template DNA is nothing but
alien piece,
Alien piece of DNA is nothing but
foreign piece of DNA,
is nothing but is nothing but template
Desired
DNA.
DNA.
Desired gene. It is nothing but desired. Who said it?
Rafa.
Rafa. Desired gene. So desired gene, alien piece of DNA, template DNA, they are all one and the same. They are all one and the same. Shall we go ahead? Shall we go ahead? Duti, Namrata,
Yes, sir.
any doubt? Any doubt?
No,
Harshavardhan,
sir.
No, sir.
Siddesh,
Yes, sir.
Sarottam,
No, sir.
Prithvi, are you there?
Yes, sir.
Prithvi? Okay. Now let us see. Origin of replication, also called replication origin, is a particular sequence in a genome at which replication is initiated. ORI, ORI. Origin of replication, also called replication origin, is a particular sequence in a DNA, in a genome means DNA, at which replication is initiated. This can either involve replication of DNA in living organisms, such as prokaryotes and eukaryotes, or that of DNA or RNA viruses, such as the double-stranded RNA viruses. DNA replication may proceed from this point bidirectionally or unidirectionally. If at all, I say that this is the origin of replication. ORI, origin of replication, DNA replication may go in this particular direction, or in this particular direction, or in both the direction. Just remember, DNA replication may proceed from the point of origin replication bidirectionally or unidirectionally. Right? Now, the specific structure of the origin of replication varies somewhat from species to species. What is this origin of replication site? The specific structure of the origin of replication varies somewhat from species to species, but all share some common characteristic such as high AT content. What is that AT content? Adenine and thymine. Repeats of adenine thymine are easier to separate because their base stacking interactions are not as strong as guanine and cytosine. So what you should know, origin of replication site, whatever you have seen, it is with the high AT content. That's all you remember. Why? What is the advantage of high AT content? They can be easily separable. The origin of replication binds to the pre-replication complex, a protein complex that recognizes, unwinds, and begins to copy. Leave it. Okay. Now let us take a further. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. Who did it? When? Where? And how? We are looking into the first case of recombinant DNA technology. The construction of the first recombinant DNA. What do you mean by recombinant DNA technology? Who will recall? Who will recall what is recombinant DNA technology? Hello? Who will answer?
desirable genes are isolated, cloned, and then transferred to target organism.
Okay, that is the DNA technology. So first attempt of that, whatever the DNA is required, isolation of that, cloning of that, and adding to the host organism, the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosome DNA of a Salmonella typhimurium. In which animal this is tried? This is tried in Salmonella typhimurium. What is that Salmonella typhimurium? What is that?
Typhoid.
Pardon?
It causes typhoid.
Salmonella typhimurium. Is it a virus, bacteria, fungi?
Bacteria.
Bacteria.
Bacteria. So first construction of the recombinant DNA technology, or first time whatever that recombination technology tried, here what is done? In this particular attempt, a gene encoding antibiotic resistance, was linked with a plasmid. Is linked to a plasmid. What is a plasmid? Plasmid is a what? Circular extra chromosomal DNA. What do you mean by this? What do you mean by this? Autonomically replicating circular extra chromosomal DNA. What is extra? Is it extra?
Self-replicating.
Jyoti,
Yes, sir.
what do you mean by this extra chromosomal DNA? Namrata?
It can replicate separately, sir.
No, I am asking you, what is extra chromosome DNA? What is the meaning of extra chromosome DNA? Navya,
Yes, sir.
what is extra chromosome DNA? Vishwas? Prithvi,
Yes, sir.
what is extra chromosome DNA? Anyone? Blessy? Rakesh?
Yes, sir.
Yeah, come on. What is extra chromosome DNA? Rafa? Aisha? Remember, Kanisa?
Don't know, sir.
Extra chromosome DNA means whenever the DNA is present outside the chromosome, it is called extra chromosome DNA. Extra means here outside. Outside. In which other organelle you come across extra chromosome DNA? In what all parts of a chromosome, or what all parts of a cell you come across the DNA? Is it that the DNA is present only in the chromosome of a nucleus? Rafa?
Sir, I didn't understand your question.
You have DNA, no?
Yes, sir.
Where you come across the DNA?
Chromosome.
In a cell, you come across the DNA inside chromosome. You know one chromosome has how many chromatids? One chromosome has how many chromatids? Hello?
4.
One chromosome has how many chromatids?
2.
2.
In which stage? In which stage? Basic point. Very good. Good that I have taken up this question. Question open to all. My question is that one chromosome has how many chromatids? And when? Can you say 2? Hello?
Yes, sir. 2 chromatin.
Who said 2?
Duty.
Jyoti, when you have 2 chromatid, where you have 2 chromatid? Namrata, Tanusri, Jayasri, Savitri, Kavita, are you there? Kavita, Kavi,
Yes, sir.
are you there? Now, who
Yes,
will
sir.
answer my question? Kanisa, Navya, Blessy, Aisha, Blessy, Rafa. Please, 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 if at all you commit mistake, I will give a margin to that. Don't worry.
G1 phase,
G1 phase how many?
2 chromatids. 1.
Who is answering this question? Hello.
Sir, in G1 it has 1 chromatid.
Who is answering this? Okay, I'll come to so that I can see. I can't see your blinking of your name, or I can't see that even that your. Okay, come on. Yeah. Who is answering this question? Yeah, G1.
2 chromatids.
Canisa. If G1 has 2 chromatid, S space has how many chromatids?
4. 4.
4. G2 has how many chromatids?
4.
4. Prophase has how many chromatids?
4.
Metaphase how many chromatids?
4.
You have written chromosome diagram, am I right? Do you whether this is taught to you in the last year? Cell, the unit of life chapter? I am asking you another question. Who will answer this question? What is the best time to study the chromosome structure?
Metaphase.
Metaphase chromosome has how many chromatids you have written? You have written a diagram of metaphase chromosome? Hello, Kanisa?
Yes, sir.
Have you written the metaphase chromosome diagram?
No, sir.
You have not written?
No.
Metaphase chromosome has 2. How many? You said metaphase chromosome has 4 chromatids, am I right?
Yes, sir.
But is it that Kanisa only interested? Is there any other students are interested in this? Please remember, you must make a correction yourself in your knowledge. In G1, anyone else you want to add to my point? What Kanisa said is wrong. Come on. Anyone has any other explanation? Anyone has any other explanation? Navya, why that you are sitting quiet nowadays? Are you angry with me?
No, sir.
And then tell me, Chinmay, Charan, hello. Anyone else has any other answer now? Nadira, do you have any other? Huh?
Sir, they align on the equator.
No, that is true. That is true. I am asking you, do you know anything about this? How many chromatids in a chromosome? So please take down, please note down, chromatid number in a G1 phase. What is that? A G1, yes, a G2? What are these 3 are together called?
Interphase.
Interphase. Interphase has 3 substages or 3 stages, G1, yes, G2. G1 phase, G1 phase has 1 chromosome, 1 chromatid. 1 chromosome, 1 chromatid. S phase, S phase, S phase, 1 chromosome, 2 chromatids. Why? Why, Kanisa? Why, Kanisa? Kanisa, why? Kanisa, why?
Don't know,
Rafa?
sir.
Huh? Rafa? Navya? What? Yeah. Have you not studied
Yes,
that?
sir.
Hello?
Yeah, sir.
Tell me, Navya, what is the importance, what is the significance of S stage of interface?
Sir, like the cell, the ones that replicates.
Huh?
DNA replicates.
You read it now, Kanisa. You said you don't know.
No, sir.
DNA replication takes place in a S stage. This DNA replication results in duplication of the chromosome. So in the G1 stage, there is one chromatid. In S stage,
Two chromatids.
two
Two
chromatids.
chromatids.
How many chromosomes, Nadira?
One
Nadira?
chromosome.
One
One chromosome, two chromatids.
one chromosome, two chromatids. G1 phase, Siddesh. G1 phase,
One chromosome, one chromatid.
one chromosome, one chromatid. S phase, one chromosome, two chromatids. G2 phase, Vishwas.
Two
G2
chromatids.
phase, G2 phase,
Two chrome.
huh? Kanisa? G2 phase,
Two chromatids.
how many chromosomes?
One chromosome.
One chromosome, two chromatids. Prophase,
One chromosome, two chromatids.
metaphase,
One chromosome, two chromatids.
anaphase,
One chromatid.
one chromosome, one chromatid. Am I right? Why? Why? Why Namrata? Why Namrata? Are you seeing me? Are you seeing me?
Yes, sir.
Yeah, okay. Of course, I am at my farmhouse. I have not brought it at a digital board. That is why I'm not using the pen. I'm not in position to write. This is G1. This is G1. This is
Yes.
yes. This is
G2.
G2. This is
Prophase.
this is
Anaphase.
Metaphase.
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 this is
Anaphase.
Anaphase.
what happened? What happened? One
Towards
chromatid,
opposite pole.
one sister chromatid. Now it has become a separate daughter chromosome and started moving to one pole. Another started moving to another pole. Telophase,
One chromatid.
one chromosome,
One chromatid.
one chromatid. G1,
One chromosome, one chromatid.
yes, sir.
One
One
chromosome.
chromosome, two chromatids.
Why two chromatids?
DNA replication.
DNA replicated, resulted in a duplication of the chromosome. Understood? Understood? So, therefore, at a given time, now I am coming back to the point. Back to the point. What is that point? Yeah. Now, the construction of first recombinant DNA emerged from the possibility of linking a gene encoding antibody resistance with a plasmid. I was telling about the extra chromosome DNA. What do you mean by extra chromosome DNA? Extra chromosome DNA is the DNA that is present outside the chromosome. Where you come across the DNA outside the chromosome? Outside the chromosome, DNA is present inside the mitochondria. DNA is present inside the plasmid. DNA is also present inside the another cell organelle. Another cell organelle.
Chloroplast.
Chloroplast. So here in the plasmid, you come across autonomously replicating. What do you mean by autonomously replicating? What do you mean by autonomously replicating? Autonomously replicating means it replicates on its own. It is not dependent on DNA present inside the chromosome. It is not dependent on chromosome DNA. It is autonomously replicating circular DNA, not this ladder type DNA. It is a circular DNA, ring-like DNA, extra chromosome DNA. I hope you understand this point.
Yes.
Generally, we write a plasmid like this. Am I right? How we write plasmid? How we write plasmid? Yes? How we write plasmid? Circle. Am I right? Ring-like. Am I right? Am I right? We write plasmid ring like, am I right? Hello?
Yes, sir.
Sir.
Yeah. So the construction first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid of Salmonella typhimurium. Where you come across Salmonella typhimurium? Where is this Salmonella typhimurium? For your information. Here, this is about cosmid, leave it. Okay, now Salmonella typhimurium causes gastroenteritis in humans and other mammals. Right? When the bacterial cells enter epithelial cells lining intestine, they cause host cell ruffling, okay, which temporarily damages the microvilli on the surface of the cell. This causes a rush of white blood cells into the mucus, which throws up the ratio between absorption and secretion and leads to diarrhea. In mice, Salmonella typhimurium causes symptoms resembling typhoid fever in humans. Right? Studying this bacteria in mice could eventually lead to a typhoid vaccine. You must remember that it is present in the intestine. Right. Now let us look into who has done this first recombinant DNA technology. Cohen had developed the technique of taking out and reinserting plasmid. Two scientists got involved. One scientist got expertise. What expertise he got? He developed the technique of taking out and reinserting plasmid, taking out the plasmid and reinserting the plasmid into another organism. Taking the plasmid from one, reinserting to another organism. Boyer had isolated a couple of restriction enzymes of Escherichia coli. So, Cohen had developed a technique of taking out and reinserting the plasmid. Boyer had developed a technique for Boyer had isolated a couple of restriction enzymes of E. coli, Escherichia coli. Both combined their technique and laid the foundation for modern technology. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. What it was doing? It was showing resistance to antibiotic. We call antibiotic resistance. Resistance to antibiotic. I hope you know. What is antibiotic? What is antibiotic? Why we use antibiotic? Antibiotic, is it antibiotic that is against our life? Antibiotic is actually against the life of most of the bacteria. So here, Cohen and Boyer accomplished recombinant DNA technology by isolating a gene which showed resistance to antibiotic by cutting out the piece of DNA from a plasmid which showed resistance to antibiotic, which was responsible for conferring antibiotic resistance. They wanted to separate that gene and wanted to insert that gene into another organism. Okay, this is what they have done. So now let us see what they have done. Cutting of DNA at a specific location became possible with the discovery of the so-called molecular scissor, also known as restriction enzyme. What is the function of restriction enzyme? Are you following me? Are you following me what I am telling now?
Yes, sir.
Okay. Two scientists came into picture. What was the expertise of one scientist? Expertise of one scientist was to just removing the DNA from one plasmid and introducing it to another. What is the expertise of another scientist? Another scientist prepared the scissor to just cut the required DNA. So, who are these scientists? One is Cohen. What is the expertise with Cohen? Cohen knew the technique to take out the plasmid and reinserting of the plasmid. He developed the technique of taking out and reinserting plasmid. Okay, another scientist, he had the technique, or he had the expertise in isolating a restriction enzyme, which is he has done in Escherichia coli. So we now got to two scientists. One is expert in taking out and inserting, another is expert in cutting. Whatever there is desired, what is that is required. The cutting of DNA at a specific location became possible with the discovery of a scissor, so-called molecular scissor restriction enzymes. The cut piece of DNA was then linked with the plasmid of DNA. The cut piece of DNA, then it was added to the plasmid DNA. This plasmid DNA act as a vector to transfer the piece of DNA attached to it. Okay? You probably know that mosquitoes act as an insect vector to transfer malarial parasite into human body. What this mosquito does? It carries. What it carries? Let us see that the mosquito is responsible for malaria. What it does? Anaphylaxis mosquito, what it does? Who will answer?
It carries sporozoites.
It carries a sporozoite from infected person to healthy person, right? So malaria parasite this Anaphylaxis mosquito is vector. In the same way, what is done here? A cut piece of DNA is added to a plasmid. Prior to that, whatever plasmid is required, that is taken out. To this plasmid, cut piece of DNA is added, and then this plasmid is inserted where? Inserted where? Inserted to where? Introduced to where? Hello.
Host organism.
Host organism. So you probably know that mosquito. Okay. In the same way, a plasmid can be used as vector to deliver an alien piece of DNA into the host organism. Is it clear, Tanusri?
Yes, sir.
Anyone wants any explanation or repetition of the same concept? Harshavardhan?
Yes, sir.
Understood?
Yes, sir.
Prithvi?
Yes, sir.
Namrata?
Yes, sir.
Lohit?
Yes, sir.
Shall I go ahead?
Yes.
Okay. Look here now. The linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase. What is a DNA ligase? DNA ligase is the enzyme required to join the cut ends, which acts on cut to DNA molecule and joins their ends. So what is the enzyme required to cut? What do you call that enzyme?
Restriction enzyme.
Restriction enzyme. What enzyme is used to join?
DNA ligase.
DNA ligase.
It's a new combination of circular, autonomously replicated DNA created in vitro and is known as recombinant DNA. Recombinant DNA. Now let me tell you, you have one plasmid like this, plasmid, and here it is cut. Here it is cut. Okay. Now it is added another DNA. It is cut here with the help of restrictions there. One more DNA is joined here, added, and it is completed. So now what we call this DNA? What do you call this DNA? Recombinant DNA. What is there in a recombinant DNA? In a recombinant DNA, you would come across one alien DNA, another. In a recombinant DNA, you would come across two DNA, am I right? Two DNA, am I right?
Plasmid DNA.
Yeah, one plasmid DNA and another one desired gene. Desired gene. of alien DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. What is done here? Salmonella DNA here. So when this DNA is transferred, from which bacteria to which bacteria it was transferred? From where we have taken the alien DNA? From which organism?
Salmonella typhimurium.
From Salmonella typhimurium, we have added it to which particular organism? Which is the host one?
E. coli.
E. coli. And we have transferred what from Salmonella to E. coli? What is the vector? Plasmid. So please remember, in this particular case, a small piece of DNA first incorporated into plasmid. After incorporating the plasmid, this plasmid got transferred from Salmonella typhimurium to Escherichia coli. I hope it is clear to you all. Everyone, is it clear? Clear?
Yes, sir.
Okay.
Yes, sir.
The ability to multiply copies of antibody-resistant gene in E. coli was called cloning. What is cloning? It is a multiplication. The ability to multiply copies of antibody-resistant gene in E. coli was called cloning of antibody-resistant gene in E. coli, because we have added the gene of interest. Gene of interest is a desired gene. We have added gene of interest from Salmonella typhimurium to E. coli. After adding to the E. coli, we want in the E. coli that get into multiple copies, and it's called cloning. The organism having a new gene or genetic material is called transgenic or genetically modified organism. Okay, Jayasri, I want you to answer this question. Which is the genetically modified organism here? In this case?
E. coli.
E. coli. Why it is E. coli is a genetically modified organism? Tanusri?
Yes, sir.
Why it is called a GMO? Savitri? Why it is called a genetically modified organism? Why E. coli is called a genetically modified organism?
Organism.
No, I can't hear you properly. Kavita? Blessy? Blessy, are you there? Now tell me. Blessy?
Sir, the question.
I don't repeat the question. Who will answer the question? Aisha?
Sir, because a desired gene was introduced
Into the E. coli. Because E. coli got a desired gene there. So it is called GMO. Blessy, you are not there. Sorry.
There is
Blessy?
some network problem, sir, here.
Your mom is there?
No, sir.
Your mom is there? Huh?
No, she is out.
Hello? Who is there with you?
Hello, sir.
Please be attentive in the classroom. Okay, I don't doubt you, but now that since you have not given the answer, I have to doubt. Okay, right. Now, you can hence infer that there are three basic steps in genetically modifying organism. 1, identification of DNA with the desirable gene. 2, introduction of the identified DNA into the host. 3, maintenance of introduced DNA into the host and transfer DNA to its progeny. Understood? These are the three important steps, basic steps in the genetically modifying organism or in getting GMO. Right? Shall we continue? Tools of recombinant DNA technology. What is that? Recombinant DNA technology or genetic engineering is the field of technology that specializes in developing new combination genetic material called recombinant or DNA, rDNA. You must know that the recombinant DNA is also called rDNA, which are artificially constructed in the laboratory for introduction in host cells for propagation and multiplication. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tool. Always ask a question, which are the tools required for DNA or recombinant DNA technology? One, restriction enzyme. Two, polymerase enzymes. Three, ligases. Four, vectors. Five, host organisms. Rakesh, Rakesh, Rakesh,
Yes, sir.
Rakesh. Why restriction enzymes are required?
Yes, sir.
Why restriction enzymes are
To cut the
required?
DNA. To
Okay,
cut the
good.
DNA.
Siddesh, Siddesh, Siddesh,
Yes,
Siddesh,
sir.
why ligases are required? Why
Sir,
ligases are required?
to join the ends.
Good. Charan, why vectors are required? Why vectors are required? Charan, why vectors are required?
To transfer, sir.
Charan, Charan, Charan,
To transfer, sir.
to transfer the gene of interest. Why host organ is required? Harshavardhan, Harshavardhan, why host organ is required? Vismita,
Yes, sir.
Vismita, why host organ is required?
Sir, for the process of replication.
For the
Process of replication.
for getting GMO. Am I right?
Yes, sir.
Yeah. So let us try to understand this in detail. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. Very important. In 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. What? One of these is methylase. One of these is methylase added methyl groups to DNA. Two enzymes in your NCERT book, they have not mentioned the details about that. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these is methylase. One of these added methyl group to DNA. They have not mentioned the enzyme name in NCERT book. Methylase added methyl group to DNA. The enzyme adds methyl group to a particular palindromic region of bacterial DNA so that restriction enzyme is unable to harm the bacterial genome. Why this methyl group is added? To see that enzyme is not harming the bacterial genome, while the other cut DNA. The one which cuts DNA is called restriction endonuclease. So two enzymes got isolated. One is methylase, which takes care with regard to the bacterial genome, and another one is restriction endonuclease. And now let us see with the restriction endonuclease. Restriction enzyme is also called restriction endonuclease or REN. It is also called simply restriction enzyme. The first restriction endonuclease, the first one, restriction endonuclease, what is this? What is this? HIND2. HIND2 from Haemophilus influenzae. Haemophilus influenzae, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. So always ask a question, which one is the first restriction enzyme to be isolated? What is it called? First restriction endonuclease is called HIND2. And it is obtained from where? It is obtained from which organism? HIND2 was isolated from Haemophilus influenzae. Okay. Haemophilus influenzae, whose function depended on a specific DNA nucleotide sequence, was isolated and characterized by 5 years later. I'll tell you, what is this specific DNA nucleotide sequence? I'll tell you about that. It was found that HindII always cut DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. Where it was, this particular, what you call this restriction enzyme, otherwise, Nadira? Restriction enzyme is also called
Endonuclease.
restriction enzyme is also called who will tell? Molecular scissor. It was found that HindII always cut a DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. What is that? This specific base sequence is known as the recognition sequence or recognition site for HindII. What is this recognition site for HindII? It is from 5' end GTCGAC. 5 to 3' GTCGAC or 5' to 3' and 3' to 5', it is a CAG-CTG. GTCGAC, GTCGAC. So where this HindII work? HindII restriction enzyme works or cuts DNA if at all it has base sequence. What base sequence should be there? What base sequence should be there? Hello? What base sequence should be there? Duty?
Yes, sir.
Where it will cut? HindII? Hello? Duty? Have you followed me?
Sir,
Did you understand? HindII. This, what is this enzyme? HindII. It is a restriction endonuclease, or simply it is a restriction enzyme. This HindII, this enzyme cuts a DNA only at the region where that DNA has what nitrogen bases? GTC, GAC. Only here it will cut. And what we call this a site? We call this as a recognition site or recognition sequence for HindII. So HindII enzyme can only cut in this. I don't know whether you have followed it or not. I shall stop it here and I will continue in the next class the same concept. Okay? Understood? Clear? So biotechnology, you must try to understand better because a lot of questions are coming from this part. Right? Okay? See you.